Today we will be joined by Dr. Mark Hughes from the University of Florida. Dr. Hughes will discuss the biology, ecology, and management of Laurel Wilt and the redbay Ambrosia beetle. This webinar is designed to help woodland owners, foresters, researchers, and concerned citizens to learn how to identify Laurel Wilt disease in the forest and residential landscape and identify potential management options. The title of Dr. Hughes' webinar is Biology, Ecology, and Management of Laurel Wilt in the Southeastern U.S. And with that, I will turn it over to Dr. Hughes.
Okay, hello everybody. I appreciate this opportunity to talk about Laurel Wilt. So let's just go ahead and get started. And if you can't hear me, just type it in the chat box so I can know. So the first thing I'd like to give is just an overview of the presentation. Basically, what is Laurel Wilt and what does it look like? How can you identify it in the field if you see it? Then I would like to talk about Ambrosia beetles and fungi in general to help clear up some confusion, and then also about the specific Ambrosia beetle and fungus that are causing Laurel Wilt disease. We'll go into the disease biology, how it spreads, where it's at right now, and then we can talk about some of the management options. And at the very end, there'll be a slide that you can find resources on who to contact about Laurel Wilt or where to get further information about the disease. So the first thing is, what is Laurel Wilt and what does it look like? Well, what Laurel Wilt is it's a tree disease that kills redbay and other members of the laurel family. So the laurel family is called the Lauricola, and Laurel Wilt is confined to trees in that family. So anything else, oaks or pines, is not a host for Laurel Wilt. It's a disease that's transmitted by a very tiny Ambrosia beetle, and it's called the redbay Ambrosia beetle. And sometimes in this presentation you'll see it as RAB for short. Now this beetle carries a fungal pathogen. So when you think of Laurel Wilt, and I want you to think of the beetle and the fungal pathogen as a team, because they're always together, and without the both of them, you can't get this Laurel Wilt disease. And the disease itself has led to the death of millions of trees, so that's why it's important. That's why we're talking about it right now. So just to quickly go into the impacts of Laurel Wilt disease, there's been some studies done where they monitor the mortality of trees in plots in certain coastal forests. And as you could see, let me get my pointer. It's anywhere from 90 to 100% of these large trees will die due to Laurel Wilt. So it's a very devastating disease to forests that are infected. And also, avocado is a member of the Lauricola, and that's now being affected in South Florida, where thousands of trees are dying in the groves. I'm not going to talk about avocado today because that is more of an agricultural disease in South Florida, and I wanted to keep this more based on forestry. So when you have this many trees dying in a forest, it's not just that you lose redbay trees or sassafras trees. There's a slew of ecological impacts that are attached to the loss of these species. One is they found that just the composition of the forest itself changes over time as you lose these large trees. And when you lose the large trees, you lose a lot of the seed source. And there is seedlings out there and resprouts, but the fate of them is still unknown. Can they get large enough to produce seed in the future? That's still something that's up in the air. Sassafras is another host, and as redbay is leaving the area predominantly with redbay, it will move into the area that is occupied by sassafras, and that's a new kind of jump in the range that we don't know how far it will go into the range of sassafras, so that's another unknown. There's a butterfly called the Palamedes swallowtail butterfly that relies on redbay as a host, and now that it's being decimated, we're seeing that their populations are falling too. So again, a lot of radiating effects. California laurel is a member of the Lauricola that lives in California, or is native to California, and we know that's a host through artificial inoculations. And then we also know that California grows more avocado than Florida. So again, if this disease was to make it to the West Coast, there's already 2 known hosts, or if it moves to the West Coast, there's 2 known hosts that it can already infest. And then lastly, just tribal cultures and medicine. In Florida, you have the Seminoles and the Miccosukee Indians, and they highly prize redbay as one of their culturally significant trees. So not only is there ecological impact, but there is a human aspect involved too. So I'm just going to quickly go over the disease cycle so that you can have an idea of how this disease spreads, and then I'll come back to it later. But right now, this is kind of just a brief overview. So what you have is, I'm going to try to pull up a pointer here, is this female beetle that will emerge from a tree, and it flies, and it's attracted to odors given off by the tree, by the leaves or the wood, and generally it will bore into the tree, and at that point the beetle has inoculated the tree, has inserted the fungus into the vascular system of the tree. And then over time, it could be a few weeks to a few months, but it's known as a rapid disease, you'll get this partial crown wilt. So you'll get large sections of the canopy that begin to wilt and turn brown. Over time, this crown wilt will spread to the entire tree, and basically what you're left with is a dead standing tree, lots of brown attached leaves, and at this point it becomes attractive not only to other redbay ambrosia beetles, but all the native ambrosia beetles that in their area are attracted to dead and dying trees. So it'll get colonized by lots of beetles, and if you're lucky, you will see these little tubes coming out. They're called frass tubes, and that's an indicator that the beetles are attacking and boring into these trees. They don't last that long because they're very delicate and it can easily be blown away by wind or rain. But again, it's an indicator that the trees are getting attacked. And once the beetles bore in, they'll make tunnels within the wood, and we call those galleries. And then they will propagate the fungus in those tunnels, and that's where the eggs and the larvae feed off of. Ambrosia beetles have a symbiotic relationship with their fungus. So you need both of them to, they need each other to survive, and you need both of them to have Laurel Wilt disease. Once the females emerge from the galleries, they'll fly off to attack new trees. So that's the basics of the disease cycle. So what are the main symptoms? I wanted to talk in generalities, and one is this transition from green to brown foliage. Wilt itself, it's called Laurel wilt, so wilt is the major symptom that you'll see on the outside of the tree. And it will, a lot of times it will keep these leaves for quite a while, depending on the host, and we'll talk about that later. But it will keep the leaves for quite a while, and then as the tree starts to get taken over by decay fungi, it will drop the leaves, and a lot of times the trees will snap in half due to their loss of structural integrity. Within the wood itself, you can see accumulation of boring dust at the base of the tree, which is, again, is an indication of the ambrosia beetle attack. Now, this is kind of a general symptom of the ambrosia beetle attack. It's not necessarily only the redbay ambrosia beetle, but if you see a redbay with a wilted canopy and this boring dust, it's a pretty good chance that you have Laurel wilt. If you were to take a hatchet and chip off the bark, you'll likely see these brown black streaks in the wood, which is an indication of fungal infection. And with the holes themselves, something I've noticed with ambrosia beetle holes is they're perfectly circular holes. So that's another, again, indication of ambrosia beetles. And if you're lucky enough, you know, the silver bullet is actually seeing the redbay ambrosia beetle itself. And I'll go over what it looks like so you can possibly identify it. So foliar symptoms. Well, one of the first things you'll see is this transition from green to brown, and it goes through many stages of, you know, green to olive. Sometimes it even gets, there's a little bit of a purplish shade in there, especially in the mid vein, kind of right there, you might be able to see it. And then you end up with these brown, crispy leaves all over the whole canopy. Here is showing kind of those preliminary wilt symptoms where you still have green leaves, but you can see that there's a loss of all structural integrity in these leaves. They're drooping. Water movement is not occurring that well anymore, and the leaves are being stressed, the tree is being stressed out, and that's showing that generalized wilt symptom. So this is we have a research farm in Citra, Florida, and we do a lot of screening for resistance. So this is just showing some plants that we have in the ground, and it's very obvious in the newest growth. In the new growth, the leaves haven't hardened off yet. They're still very fleshy and soft, and you'll tend to see laurel wilt a lot easier on this new growth. You'll get these wilting leaves, so oftentimes they curl, sometimes they even scorch. And it doesn't even necessarily have to be on the top of the tree like here. A little lower you can see this new growth. It generally is a lot lighter in color. That's another indicator of laurel wilt. So crown wilt. Going from those preliminary symptoms, what you end up getting is discoloration and wilt, but I want to emphasize that it occurs in large chunks of the canopy at once. Branches. It's not a twig or a little piece here and there. It's generally large pieces of the tree in sequence. So I'm kind of pointing over. You can see that they start to yellow, discolor a little bit here, and then you get, again, this large chunk of wilt. It will eventually progress to the whole entire tree, and then you're left with these dead standing trees. So this is kind of worst case scenario. You can see that in these residential areas, I use a lot of residential photos because it's hard to get photos like this in the forest just because of the background, other things competing with it. But here you can see these large standing trees, full of wilt, and at this time the tree's completely dead. It's possible they can resprout from the base, and that does happen sometimes, but the main tree is a goner. And you can see over here how it's progressing down the line, and you can see a tree that's about half wilted right here. And this is likely due to beetle movement. We don't think root graft transmission is happening in kind of the residential areas. There's different hosts that it's more likely, but there's no real indicator that it's happening right now due to underground transmission, at least in the redbay system. So this is just to help out a little bit, wilt or not. Now sometimes we get calls or people think that they have laurel wilt when they have these small twigs sporadically throughout the crown that is causing wilt. And this is actually something called black twig borer. And this is caused by another ambrosia beetle, and it attacks lots of hosts. It's a nursery pest. It's a pest of coffee in Hawaii. Has a wide host range, but it's mostly a twig killer, individual twig killer. It does not cause a systemic disease. So if you see just individual twigs sporadically through the crown, my first guess would be look for black twig borer. Again, with laurel wilt, it's systemic. You get effects on the whole crown itself. So just to talk about the black twig borer, it is often misdiagnosed as laurel wilt. It's caused by another ambrosia beetle. And again, if you look at number B, or letter B, it infects single twigs at once, so you're not going to get the systemic wilt. And one way to check is if you cut that branch off, or that twig off, and you look really closely, most of the time you'll find a little small hole. Again, perfectly circular hole. And if you were to snap that in half, a lot of times you'll see the beetle in there with eggs, immatures. And this is all they do. They colonize this little area. They'll kill that little twig. Sometimes they can kill small branches, but it's not a tree killer. So I just want to be clear on how those two can kind of be confused and help you clear up the difference between the two. And a lot of times you can get a redbay that has black twig borer damage, but also has laurel wilt. And the way to differentiate is look on those individual stems, look for the boring holes, and then just look at the distribution. Is it more systemic, or is it just a scattered branch here or there? So at the end, after the tree has been fully colonized by the laurel wilt fungus and it's died, what happens is you start to get a lot of decay from other decay fungi that will take over the tree. and you'll get this loss of structural integrity. You know, it can't hold that crown up anymore. So you get these trees that are snapping, and those are all the ones that are outlined in red. So maybe a few years after these trees have died, you go back to a site that's been heavily infested by laurel wilt in the past, and you'll see just a lot of these dead and standing trees. There's no integrity left. You can push these trees over, you know, by yourself if you wanted to. Something else to note is if you see any decay conchs or anything like that, that's not the laurel wilt fungus. That's usually the decay fungi that have colonized this tree. There's all different types, so I just want to make that known that any type of conchs or anything you see is not the laurel wilt fungus. The laurel wilt fungus is not a good saprophyte, so it means it's not a good decay fungi. It survives well within the galleries when the tree has very little competition. But after it's died, generally the laurel wilt fungus will get outcompeted by other organisms. So now we'll move on to the wood symptoms. So again, if you took a hatchet and you were able to chip off the bark, this is the main difference you'll see. Healthy wood is nice and white, cream colored, and an infected tree will have this vascular discoloration. And this is an indication of fungal infection by the tree. Lots of vascular wilt diseases have this type of internal streaking. So that's, you know, one of the more ways you can be confident that you have laurel wilt. And sometimes you have to chip around in a few different locations at once because you might not hit a spot that has that vascular discoloration. So I would suggest trying a few little areas to find that discoloration if you don't find it initially. Okay, another indication again is Ambrosia Beetle boring. So here on the letter A, you see these, again, these are those little frass tubes. As the beetles bore in, they push out the sawdust behind them, and it comes out in this very nicely formed tube, and we call them frass tubes or frass toothpicks. And again, those don't last that long. If a windstorm comes or rain, it will blow them away. And more likely, you'll see B, which is accumulated frass at the base of the tree. So when you see this is a good indicator of high number of attacks. Those initial attacks that actually infect the tree a lot of times are undetectable and they're hard to find because you don't need that many beetles to inoculate a tree and start the disease process. Unfortunately, the fungal pathogen is very virulent, so you don't need that many beetles to actually kill it. We did an experiment, and we found that within 100 spores is able to induce wilt and death. So by the time you see these type of symptoms, your tree is already showing, it's already showing wilt. It's already on its way out, unfortunately. But this is a good idea just to know that you have Ambrosia Beetle attack. Okay, so now we'll go into the redbay Ambrosia Beetle itself. So the vector, the insect that transmits this disease is called the redbay Ambrosia Beetle, and it's Xyleborus glabratus. That is the species name. And if you were to cut a tree in half and look at a cross-section, what you'll see is all these little galleries, and these are the tunnel systems that they make within the wood. And that's where they lay their eggs, and that's where their young feed on the fungus within the gallery walls, and they generally spend the majority of their life within these trees. They generally only leave to go find a new tree to infect. So controlling the insect itself is a hard task to do. Again, you can see these galleries. There's the fungus. This is not the laurel wilt fungus actually within it. It's likely another decay fungi that has colonized these tunnels. Letter B, you can see the larvae developing here, and this white fuzz is actually that symbiotic fungus. And how that works is that the fungus, the wood tissue itself there's not very little nutrition for these insects. Unlike bark beetles that will feed on kind of underneath that bark, that vascular cambium area and the phloem, that's very nutritious, and they can get nutrition out of that. Ambrosia beetles bore through that and establish themselves within the wood. So what they have to do is rely on this their symbiotic fungi to kind of ramify into the wood and extract nutrients and then bring it back to that gallery. And that's what they feed on. So again, when you think of the redbay ambrosia beetle, think of its fungal partner too. So this is just another illustration of their gallery systems. You can see right here they enter, and again, most bark beetles hang out in this area, but they'll go in deeper, and this is where their galleries will be occupied by their symbiotic fungi. This is where they'll lay their eggs, and this is where they spend the majority of their life. So now I'm going to talk a little bit about just ambrosia beetles in general and fungi. And I want to do this just in case some people aren't familiar with ambrosia beetles or hear of ambrosia beetles and bark beetles and what's the difference. So let me try to explain that right now. So ambrosia beetles are tiny wood-boring beetles. On the right, you can see there's all different species. Unfortunately, unless you're an entomologist, they can kind of all look the same in some ways. Ambrosia beetles are dependent on a fungal partner. Again, they rely on their symbiotic fungi for survival. They live in excavate tunnels in the wood, and again, those are called galleries. There's thousands of species in the United States. Most of them are benign. They don't really cause any impacts because they colonize weak and dying trees. So most of them are good for the environment. They help with nutrient cycling. They help kill off trees that are already weak and stressed. So all ambrosia beetles are not bad. So what's the difference between an ambrosia beetle and a bark beetle? I mean, there's lots of differences, but if I was to boil it down, it would be the galleries and the way their orientation in the wood. Again, with an ambrosia beetle, it goes kind of horizontally, deep within the wood, where a bark beetle will more feed kind of in that layer just underneath the bark. So their galleries kind of go more vertically because they're not interested of the deep wood. So that's one indication. So if you were to remove the bark of a tree and you see all these squiggly lines and galleries, that's not an ambrosia beetle. You have to cut deep within the tree to really see those galleries of an ambrosia beetle. Ambrosia beetles, again, they have the symbiotic fungus that they need to survive. And bark beetles, they feed on this underneath the bark tissue. They also associate with fungi, but their relationships are variable depending on the species of fungi and the beetle and the condition of the tree. So with ambrosia beetles, it's a much more obligate association between the two. And just for example, the redbay ambrosia beetle is a well-known ambrosia beetle, and southern pine beetle in the south is a problematic bark beetle. So helpful or harmful? Native versus exotic ambrosia beetles. Well, again, the native ones generally attack weak and dying trees, where the exotics are a lot of times attack healthy trees. And I'll talk about what they're attracted to. Most native ambrosia beetles are generalists. Again, they are just looking for whatever stressed and dying tree that they can colonize. So a lot of them attack many different species, where a lot of these exotic ambrosia beetles tend to target specific hosts. Or, with the Laurel Wilt redbay ambrosia beetle, it's within the plant family Lauricola. Not that they're all host specific. There are some generalists. But a lot of these ones that cause disease tend to be more host specific. Of course, if it's a native ambrosia beetle, it's going to carry a native fungus with it, where these exotic ones tend to bring in their own exotic fungi with them. And a lot of these fungi are the tree killers and the pathogens, where in the native ones, they're generally not pathogens. They're just fungi that help degrade trees, bring nutrients to the young, but they're not necessarily on their own affecting or killing trees. And again, it's just balanced. It's similar to all native and exotic organisms. The native ones tend to be more in balance with the ecosystem, where these exotic ones, again, they're tree killers. They disrupt ecosystems. There's no natural enemies. So that's how you can get these exotic animals, causing a lot of damage in these forest systems. And The good thing is they tend to be more rare. With international trade, ambrosia beetles are being brought over all the time. So there's a lot of ambrosia beetles that come into the United States, but the minority of them are actually tree-killing pests. So, and the redbay ambrosia beetle and Laurel Wilt is kind of the major pest that we know of in the United States that's causing this systemic wilt, and it's killing millions and millions of trees. Okay, so with the redbay ambrosia beetle, this is what it looks like. It's a small black-brown beetle. Again, it's a little bit over a millimeter in size. Now, the female is the sex that is responsible for the transmission of the disease. What they have in them is, it's called a mycangia, and it's basically a fungal pocket. Now, their mycangia are mandibular mycangia, so they're within their heads, kind of next to their mouth parts. It's not this antenna, but it's within their mouth parts. And that's where they carry the fungi. They carry them in kind of a spore phase. And the females are the ones that are capable of flight. The males can't fly, they do not have a mycangia, and they're generally just left in the host tree for reproduction. The ratio of males to females is highly skewed toward females, so you might have 30 to 40 females produced for every male. And that, of course, can change due to hosts or environmental conditions, but in generalities, there's a lot more females than males. And here is just showing how tiny it can be. And I'm not an entomologist, but one of the easiest ways to identify the beetle compared to other native ambrosia beetles is kind of the black-brown coloration it has, and also this declivity right here, this arch. It's very flattened. You can see it on both sides. And then it's smoother than a lot of other ambrosia beetles. Glabratus, the glabra means smooth, so that's kind of a way to help out in identifying the beetle. So this is just more for illustration purposes. You could see all the life stages of the redbay ambrosia beetle, Xyleborus glabratus. It starts from a small egg that's laid in the gallery, and then it will mature to different life stages or instars. You get the pupa, and then those develop into, they're called teneral adults. So they're kind of younger adults, and they're a lot more soft and lighter. And then as they mature, they harden off, get darker, and then at this stage they're ready to fly out of the host tree to attack new trees. Here's the male, and you can see that it's a little bit smaller. Again, it's actually haploid, meaning it has only one set of chromosomes, so it's physiologically a less advanced form of the beetle. It also has little teeny horns. You can't really see it here, but that's another way to tell a male from a female. It has these little horns here. And again, those stay mostly in the natal gallery. They don't spread Laurel Wilt. They can't fly. Okay, about redbay ambrosia beetle development. Basically, the main point I'm trying to make is that an infested tree is essentially a beetle factory. You can get thousands and thousands of beetles emerging from an infested standing tree. After about 30 days, you can find all the different life stages of the beetle, from eggs to larvae to pupa to adults. So it doesn't take long for the reproduction to start happening. Egg laying is very continuous. So again, this is when it comes to management and sanitation, you want to make sure you remove as many standing trees as possible, because this is what really helps propagate the disease and propagate the beetle itself. So I just wanted to make that point. So now we'll talk about disease biology and spread. So again, hopefully this makes more sense now. I won't go through it real quick. I'll go through it real quick, just saying that, again, you have this female that just a few ticks, it's even thought that maybe one or two is enough to infect a tree. And once it's infected, you'll get that wilt, and then it's attractive to not only the redbay ambrosia beetle, but lots of native ambrosia beetles. They lay eggs in their galleries, they develop, and then they fly off. And that's the disease cycle. So what is the redbay ambrosia beetle attracted to? That's a good question. The first thing is odors. So they don't have a really good sense of sight, not like humans where they can pick out trees from afar. They rely on more these odor plumes. And one of the most powerful to them, it comes from the wood. Now there's many specific compounds they're attracted to, and they're very conserved within the family Lauricola. There's one called alpha-copine, which is used in manuka oil lures, which is a lure that's used for emerald ash borer. But at this point, there's been work done, and they've refined it even further, and they've distilled this alpha-copine and concentrated it. So they have very good lures out now to attract the beetle. In nature, wounding or exposing the wood is also a good way to increase beetle attacks, because in the trunk, even though volatiles are emitted, and volatile is just a scientific word for odor, even though they're emitted, they're kind of bound within the tree more than when you expose the wood. And if you were to expose the wood and you had redbay ambrosia beetles in the area, you'd see a lot of beetle attacks on those areas. We also know, we did some research, and even the leaf odors themselves can attract the beetle. There's a lot more, volume-wise, a lot more volatiles that are being released from the leaves. So we think that these might act as kind of more of a bit of a longer range cue, that there might be Lauricola in the general area, and then the wood volatiles will really bring them in. There's been work showing the fungus itself is attractive to the beetle. And then we also know that as they once they land on the tree, they will walk around it, and then they can also sense it and take little tastes to see if this is a host. So there's lots of steps before the beetle just bores into a tree. It wants to avoid boring into a non-host. But odors, we know, is the main driver of attraction. Something else that was shown was that visual cues do help. Infested areas or forests, generally the largest trees tend to die first due to Laurel wilt, and it's thought that that's probably a combination of more odor coming from these larger trees, but also the stem silhouette itself. There's been research done with artificial stems that have the same amount of attractant on them, and it was found that they migrate to these larger stems first. So it's probably a combination of visual and scent cues. So this is the distribution of Laurel wilt. I wasn't able to get a hold of the latest map, but there is an infection in Arkansas that just happened in late December. As you can see, the beetle was first captured in 2002 at Port Wentworth, which is right near Savannah. It was detected in some traps, and at that point nothing was really thought of it because they intercept lots of beetles in traps. But then within the next year or so, they started noticing lots of mortality of redbay, and then the Forest Service got involved, and they were able to decipher this ambrosia beetle fungus relationship. And from there, basically the disease is spread along the coast of where redbay is very prominent. So most of the spread was going north-south. And then over time, it started to just expand westward, and you get a lot of this is due to just beetle movement, but then we do have these jumps in host range or in geographic range, and that's thought to be the possibility is that it's movement of wood materials. That's been a documented case in Florida where there was somebody lost their tree due to Laurel wilt. They cut it down, left the logs on the side of their road, and then a wood turner saw those beautiful logs with beautiful stain in them, took them home to his house, put them in his garage, and within a few weeks of that, his redbay tree was dead. So movement of firewood is obviously an important thing to minimize the spread of this disease due to human factors. And also, just to let you know, if you guys think you find a detection of Laurel Wilt, Chip Bates would be the person you want to email, because he updates this map, and that's the source of how we know where it's spreading through time. So disease spread. Again, most of it, I'm showing the range of redbay and the range of sassafras. So as you can see, redbay hugs the coast, and it goes, there's a little spot in Texas, and then sassafras has a much larger host range, and that's what we're kind of worried about, is how much of this range can it cover. It's a subtropical beetle, so research is being done on the climate matching, and how far can this subtropical beetle move. Something that's not working in our favor, again, is that these beetles live deep within the tree, and the wood itself has the ability to kind of buffer the temperature. There's two modes of movement. One is natural, which again is just the beetle moving from tree to tree. It was estimated by a scientist at the Forest Service that they can move at about 55 kilometers a year. But we also know that there is long-distance flight. That was shown in the Everglades. The Everglades has these tree islands, they're called. They're individual islands that are separated by water, and they've mapped the jumps from infected island to non-infected island to infected island. So it is known that they can do some long-distance flights. Human-assisted transport is another big issue, and that's hard. It's important because it's very difficult to predict, and it can lead to these larger jumps in distribution, bringing the beetle where it wouldn't go, or at least where it would take a lot longer to reach. So now I'm going to talk about hosts. So again, it's the laurel family, it's the Lauriceae. It's also called sometimes the cinnamon family because of camphor tree. It's the cinnamomums. Oh, I'm sorry. So I just realized this is the reverse. It's actually Cinnamomum camphora. I'm sorry about that. I apologize. A lot of times what you hear is redbay, and that's a common term for a species complex. And you have redbay, swampbay, and silkbay, and they're all very similar. They just have different ecotypes, slightly different anatomical differences, but you're pretty safe if you just want to call them redbay. There's camphor tree, sassafras, bay laurel, and pond spice. Those are the ones I'm going to talk about. There's lots of other hosts within the family Lauriceae. A lot of them are either threatened or endangered, like spice bush, Gulf Lycaria, and pond spice. Where is pond spice? Oh, pond spice. And then some were just done through artificial inoculation. And again, the stress, it's the laurel family only. So you're not going to get laurel and pines, oaks, magnolias, or anything that isn't in the Lauriceae. So redbay, you've seen these earlier. Again, you're going to get that canopy wilt. This is a silkbay right here. It prefers a dune habitat. It's very limited geographic range, just to certain parts of Florida. Here's redbay. So again, just showing those symptoms. Camphor tree. So in Asia, camphor trees can get gigantic, and they're beautiful trees. It was introduced to it's an introduced tree, and it is common, at least in Florida, to the urban landscape. And what you're going to see with camphor is it's not going to cause the wilt that you're going to see in redbay. It's more of a branch dieback, where you're just going to get individual branches dying. And this is also the fruit you could see. A common thing with Lauriceae as a family is they're very aromatic. So if you were to take the leaves and crunch them in your hands and smell them, they have a very pungent smell. Camphor tree is one of the sources of Maxima, the skin cream, so it has a very aromatic smell to it. So with camphor tree, there was some work done by Forest Service scientists at in Georgia, and there is dieback involved by Ambrosia beetle attack, but it's more of just a branch dieback. Again, here you can see a little bit of the the branches dying, but you're not going to get complete tree mortality unless you have a massive amount of beetle pressure. And we think it's just because, again, since the beetle and the fungus came from Asia, possibly there's a coevolution because this host, the camphor tree, also came from Asia. So that's one reason we speculate that it's not causing this widespread mortality. And also, I mean, sometimes it's called camphor tree in one word, but that can also be two words. Something else you can notice about Laurel wilt is it could be misdiagnosed as squirrels chewing on the bark. Many times here in Florida we see a dead branch and we think it's Laurel wilt, but then upon closer inspection we'll see this chewing, and it's basically these squirrels are girdling the branch and causing the branch to die. So if you have camphor in your area and you see a branch that's wilted, take a closer look and see, is there some type of chewing or physical damage to the bark that's causing that girdling, leading to the wilt symptom. In Florida, this is not really a big issue, Laurel wilt and camphor tree. Again, it's just a little bit of branch dieback. Sassafras is another host of concern. In Florida, we don't really have much sassafras. It's very small compared to other parts of the country. But it is a medium-sized shrubby tree. It's deciduous, which means that it's going to drop its leaves after a time, and that can make it a little bit harder to diagnose. Something else is they share a common root system. A lot of times these sassafras trees, they grow in kind of rows, and they propagate through stump sprouts or through root sprouts. And it's seen that you get underground transmission of the disease. As one clump will get infected, and then they can move through that connected root system to infect the other ones. So hasn't been proven through experiments for sassafras, but there's a lot of anecdotal evidence that points to it. So you should just be aware of that. And again, it's a major host of concern just due to this gigantic range it has. Okay, so it's easier for me to just show a lot of photos of sassafras and how the disease looks in this host. So again, you're still going to get this wilt of these leaves that will again die back, get brown and crispy. With the wood, you're still going to get that same vascular discoloration, Ambrosia Beetle attack. Again, vascular discoloration within the wood, and this is the root flare, and as you can see, it infects the root system, so that's another reason why we think root graft transmission happens. And I'm just going to go through this time series here of Laurel wilt. So most of this is sassafras. It'll be easier to tell as we go through. But again, you can see how the canopy is spinning out. You're losing leaves. You're dropping leaves until you're left with this. So that's really what Laurel wilt and sassafras looks like. Now, bay laurel. Bay laurel is in the United States at least, it's an ornamental shrub or tree, and it's actually, when you think of the bay spice, bay leaves, that is bay laurel. And we found in Florida, in Gainesville, in a botanical garden where it was planted, in close proximity to an avocado that recently succumbed to Laurel wilt, we found, again, Ambrosia Beetle attack, vascular discoloration, and this wilt symptom. So it is a host to Laurel wilt disease, and again, just showing the difference between a healthy vascular system and an infected vascular system. So again, if you have bay laurel planted in your landscape, it is susceptible to the disease, and it is a host, and the beetles are attracted to it. So that's another something else you have to worry about. Pond spice is a very rare shrub. There's some in Florida, and also researchers at the Forest Service in Georgia have done some work on it. They've done the majority of the work of it. But it's deciduous, so again, and it's a multi-stem shrub, and it likes certain kind of swampy areas. This is just showing the difference between a healthy and an infected tree. And since it's smaller in stem, if you could see that gallery right there, it's more of a suboptimal host, we think, just because there's not a lot of physical room for these extensive gallery systems. But there has been research done already showing that once all these large trees die, the beetles are able to persist, the population can persist in these small diameter stems. The population numbers will crash, but they won't disappear. They can persist in these smaller stems. And this is an endangered species, so this is one that's of concern. Last is management options. So there's been some work on protection. So we know that the beetles prefer to bore into the lower portions of the trunk. Lots of trapping studies at different heights found that these lower traps attract the beetles. That's where you're going to get most of your frass from and your boring attacks. So something that was done was they just looked at protecting them with screen barriers. And what was found, a lot of research was done, hard work was done on this, but unfortunately it was found that they will just go above the barrier, or they're so tenacious they'll actually just bore through the metal screen. So unfortunately, it's not a suggested management option. Sanitation. Really, sanitation is the best management option you have right now. Just due that Laurel Wilt is a forest disease, you can't spray chemicals over the whole forest. But if you can remove the dead and dying trees, remember these dead trees are beetle factories, so if you can remove them, it's the best way you can lower the population, the beetle population. It's been tried to eradicate the disease completely from an area. It was tried in Georgia, in Jekyll Island in 2007, I believe, and a lot of physical work was put into eradicating the disease. Unfortunately, it didn't work, and Laurel Wilt popped back a few years later, but it probably bought some time. So sanitation is still a very good thing to do. And the main goal of sanitation is really to physically destroy the beetles in their habitat. The beetle itself is kind of fragile once it's outside of the gallery. And the fungus isn't that good of a competitor. So if you were to destroy the wood, you're basically destroying the house and the fungus can't grow. By promoting wood drying, you're selecting against the Laurel Wilt fungus, the Raffaelea fungus, and promoting competition. So there was a study done by wood chipping and how the beetles survived and how the fungus survived in chipped wood. So this is just a comparison. In unchipped wood, basically, for the same volume of wood, they were able to rear over 1,000 redbay ambrosia beetles over the course of 10 months. Where when they chipped that same volume of wood, they only got 10 beetles out. If they were to cover these chip piles, 0. They caught 0 beetles. Now for unchipped wood, when we left the tree standing, we were able to isolate the pathogen anywhere from 3 to 15 months after we started the experiment. But with the chipped wood, the pathogen only lasted 2 days. So again, the pathogen is it's not a good survivor outside of the protection of its gallery. And the beetles are they're fragile, so they easily can get chipped up and damaged and die. So these are just some very generic sanitation recommendations, going kind of in an order from least attractive to most attractive. One is just to cut down the tree and leave it on site. By cutting the tree down, you're separating it from the root system, which will help it dry out further, but still you can get a lot of beetles emerging from even a cut down tree left on site. Again, that chipping study before, those were logs, and they still were able to release over 1,000 beetles in 10 months. By cutting it and chopping it down, you're basically increasing the drying, which is adverse to the fungus, because there's a wood moisture range that the beetle and the fungus need to live. So by chopping it up, cutting it into smaller logs, you're increasing drying and you're increasing fungal competition. And covering it is even better, obviously. Cutting, chopping, covering it is even better. Again, we just went through chipping. That's really good. And that's kind of the ideal way you can do it. And again, all this should be left on site. It's not on there. But you really shouldn't be transporting this wood material anywhere. You should leave it on site because even the chips, even 10 emerge, so that's still enough to move the disease around. Again, by chipping and covering, there was 0%, 0 beetles emerged. So that is the ideal option. It's thought that if you don't have a chipper or any type of hardcore, wood destroying equipment, you can cut it down and take it to your municipal dump where they can basically just bury it. But that, again, is only if the county already has laurel wilt. What you don't want to do is take this. I'll get to the questions at the end. I see some questions popping up. You could take it to the dump, but again, only if it's within the county or nearby. You don't want to be transporting these logs long distances. And again, the main point is destroy the wood as much as possible. When you destroy the wood as much as you can, you're hurting the beetle and you're hurting the fungus. And don't move wood around. So there's a website called don'tmovefirewood.org, and this is a great resource with all the different wood boring. It's not just laurel wilt. There's lots of wood boring insects that can be moved around. So this will give you rules. There's laws by state. It's a good resource. I have to go through this. I'm almost done, kind of quickly. Chemical methods. So if you have a large, high-value tree that you want to protect, you can use the fungicide propiconazole. The one we tested was by the trade name Alamo. Now what it is you have to use a macro infusion process where you tap into these surface roots and then you can pump in the fungicide under pressure. And basically what that gets is that's going to help give a good even distribution through the vascular system. It should be done by a professional because it can be tricky sometimes getting these taps that they tap into to actually uptake the fungicide. It should be done on a warmer day to increase transpiration, again, to help pull the fungicide up the tree. Unfortunately, it doesn't last forever. In Florida, it's been shown to last between 12 to 18 months, and a lot of that varies on how well the uptake was by the tree. There's been some research on insecticides. They're not very effective. Again, remember, the beetle lives most of its life deep within the tree, so there's really not much of an opportunity for the contact between the insecticide and the insect. So all the work that's been done has pretty much shown that it's not a viable option. This is just a word of warning for the homeowners or other people that maybe get solicited for fungicide treatments. Standard fungicides will not work. There's nothing you can get at Lowe's or Home Depot that is going to work for laurel wilt. I've visited people and they say, oh, don't worry about it. I got something from Lowe's. I'm going to spray it on the tree. It says it kills fungus. And of course, a few months later, those trees there's just a stump of a tree. So, nothing you can get at Lowe's or Home Depot is going to work. Right now, the only thing tested has been that propiconazole through the macro infusion process. There's also other ways to get the fungicide into the tree, but the only one we really suggest is this macro infusion process. And of course, these recommendations change over time, so you want to refer to your extension agent. You can email me, and we can try to give you the best up-to-date recommendation. Host resistance. Part of my research was basically looking for resistant redbays in particular. So we'd go to areas where the disease has killed most of the trees, and we select those few remaining survivors. We propagate them, and then we grow them and plant them in a farm outside, in a research farm, and then we inoculate them, and we see who's the survivors. So we have some good tolerant redbays in development, and hopefully down the pipeline those will be available for homeowners or just for general kind of reforestation efforts. Last slide is information resources. So you can check your local forestry agency websites, the Florida Forest Service, Georgia Forestry Commission. All of these sites, all the states along the the coast that have laurel wilt generally have some information about laurel wilt disease, and you can they have contacts there. Again, you can use your county extension agent. the Southern Regional Extension Forestry webpage, which is obviously where you're at right now. We have a fact sheet that is on the way. It's being prepared maybe in about 60 to 90 days. It should be out. We also made a document for the USDA called the Laurel Wilt Recovery Plan. And that's free, and it's available in the journal called Plant Health Progress. And if you Google Plant Health Progress, Laurel Wilt Recovery Plan on redbay, it should be pretty easy to find. And it's the more extensive document of what you've seen, all the efforts of other treatment methods that haven't worked, that I really didn't mention because they didn't work. And again, there's an infrastructure and experts section at the very end that has state by state who to contact about Laurel Wilt and Forest Service contacts. I have a website, laurelwiltresearch.com. It's mostly photos and all the scientific research that's been conducted about Laurel Wilt. And then there's University of Florida has an EDIS database, which is like an electronic encyclopedia where you can type in Laurel Wilt in the search bar, and there'll be small 1 to 2 page documents about Laurel Wilt on the landscape, Laurel Wilt in the residential area. These are only a few of your information resources, but this is where I would definitely start. And with that, it's 1:59 and I'm done. So again, I want to thank you for all your time. If you had any questions, just email me. I know I didn't get to everything, but again, email me and I'll try my best to answer your questions. So from here, I'm going to turn my microphone off.
Okay, thanks, Mark. Great job. We've got a few questions rolling in. If you haven't already sent your questions, please put them in the chat window. We will get to a few of these here. The first question, Mark, and I'll ask you the question, then you can turn your mic back on and address it. Has fire, wild or prescribed, been shown to have any effect? Okay, has fire, wild or prescribed, been shown to have any effect? There hasn't been any research